Section 2. The Genetic Foundations of Development. The body contains trillions of cells. Each cell contains a cell structure, the nucleus. The nucleus of each cell contains chromosomes, thread-like structures made up of deoxyribonucleic acid, or DNA. DNA is a complex molecule that contains genetic information. Genes, the basic units of hereditary information, are short segments of DNA. These direct cells to reproduce and assemble proteins that direct the body processes. Each gene has a designated place on a particular chromosome. Completion of the Human Genome Project has led to the use of the Genomewide Association method to identify genetic variations linked to a particular disorder. To conduct a genome-wide association study, researchers obtain DNA from individuals who have the disease and individuals who don't. Then each participant's complete set of DNA or genome is purified from the blood or cells and scanned on machines to determine markers of genetic variation. If the genetic variations occur more frequently in people who have the disease, the variations point to the region in the human genome where the disease-causing problem exists. Genome-wide association studies have recently been conducted for cancer, cardiovascular disease, depression, suicide, autism, ADHD, glaucoma, and Alzheimer's disease. Linkage analysis is used to discover the location of one or more genes in relation to a marker gene whose position is already known. Genes transmitted to offspring tend to be in close proximity to each other, so that the gene or genes involved in the disease are usually located near the marker gene. Next-generation sequencing refers to a vast increase in genetic data generated at reduced cost in a shorter period of time. Using recently developed next-generation sequencing, an entire human genome can be sequenced in one day. Prior to recent improvements, deciphering the human genome took over an entire decade. The new technology sequences millions of small DNA fragments. Next-generation sequencing has considerably increased knowledge about genetic influences on development in recent years. A current project that began in 2008, the 1000 Genomes Project, aims to determine the genomic sequences of at least 1,000 individuals from different ethnic groups around the world to study genetic variations in disease. One of the big surprises of the Human Genome Project was finding out that humans only have about 20,000 genes. Before the project, scientists thought that there would be hundreds of thousands of genes because we have so much variety, and they thought that each gene programmed just one protein. What we discovered, though, is that rather than being a group of independently operating genes, genes collaborate with each other and with non-genetic factors in and outside of the body. You might have a gene for something, but it's not just about having or not having a gene. Genes can be turned on or off by things like stress, exercise, nutrition, radiation, or sleep through a process called methylation. So how is this genetic information passed down? All cells in the human body, except for the eggs and sperm, have 46 chromosomes arranged in 23 pairs. These cells reproduce through mitosis. The cell's nucleus, including the chromosomes, duplicates itself and divides. The two new cells each contain the same DNA as the original. Eggs and sperm are formed through meiosis. A cell of the testes or ovaries duplicates its chromosomes, but then divides twice, forming 4 cells. Each has only half of the genetic material of the original. When a mommy and a daddy love each other very much. During fertilization, an egg and sperm fuse to create a single cell called a zygote. The zygote has 23 single chromosomes from the egg and 23 single chromosomes from the sperm. So now it has 23 pairs of chromosomes, half from the mother and half from the father. Set A shows the chromosome structure of a male, and set B shows the chromosome structure of a female. The last pair of 23 chromosomes is in the bottom right box of each. Notice that the Y chromosome of the male is smaller than the X chromosome of the female. To obtain this kind of chromosomal picture, a cell is removed from a person's body, usually from inside the mouth. The chromosomes are stained by chemical treatment, magnified extensively, and then photographed. Combining the genes of two parents in offspring increases genetic variability in the population. This is a good thing because it provides more characteristics upon which natural selection can operate. Another source of variation is mutation, where either chance or environmental damage permanently alters segments of DNA. Sometimes this mutation can be positive, negative, or make no discernible difference in a person's function. All of a person's actual genetic material is called their genotype, but whether and how much these genes actually result in observable and measurable characteristics is called your phenotype. For example, you might have inherited a gene for brown eyes from your mom and a gene for blue eyes from your dad. That would be your genotype, one brown allele and one blue allele. But whether you actually have brown or blue eyes, that would be a question about your phenotype. Phenotypes can be physical and psychological, such as how curly your hair is or how anxious you are. There's also a range of phenotypes that can be expressed for each genotype. For example, you might have inherited genes that give you the potential to become very tall, but your level of nutrition will influence where within that potential height range you fall. Malnutrition stunts your growth. So what determines how a genotype is expressed to create a particular phenotype? It's complicated and we don't have a full understanding of it yet, but there are some genetic principles that have been discovered. One is that of dominant and recessive genes. A dominant gene overrides the expression of a recessive gene, where a recessive gene only gets expressed if both genes in the pair are recessive. Brown is the dominant eye color gene. If you inherit even one brown eye gene from a parent, then you will have brown eyes. The only way for you to have blue eyes is for all of your inherited eye color alleles to be the recessive blue. So let's say you inherited a brown-eyed gene from your mom and a blue-eyed gene from your dad. And then you have a baby with someone who also inherited one of each gene from their parents. You and your partner would both have brown eyes. Oh my gosh. You and your partner would both have brown eyes. It's because brown dominates. Okay? The brown gene dominates. But there is a 1 in 4 chance that your baby will inherit your and your partner's recessive blue-eyed gene and have blue eyes. So 2 brown-eyed people can have a blue-eyed baby. Another thing to know about are sex-linked genes. When a mutated gene is carried on the X chromosome, the result is called X-linked inheritance. This is a much bigger deal for males than for females, because females have 2 X chromosomes, so one can act as kind of a normal backup copy. However, males only have 1 X chromosome. Their other sex chromosome is a Y, so if there is damage to their Y chromosome, they don't have a backup. Another thing we know about is genetic imprinting. This occurs when genes have differing effects depending upon whether they are inherited from the mother or the father. For example, there are 2 very different syndromes, Prader-Willi syndrome and Angelman syndrome, that are both caused by the same deletion in the same spot on chromosome 15. If the deletion was on the chromosome 15 that came from the father, then you get Prader-Willi syndrome. If the deletion was on the chromosome 15 that came from the mother, then you get Angelman syndrome. A chemical process appears to be silencing one member of a gene pair, creating different effects from the deletion. Now, most human characteristics aren't determined by just one gene, though. Most characteristics are polygenetically determined, meaning they are determined by the interaction of several genes. When the Human Genome Project began, scientists were hoping that we'd be able to simply find things like the Alzheimer's gene or the type 2 diabetes gene. But it's not as simple as that. Multiple genes, along with environmental factors, are influencing our characteristics, behaviors, diseases, and development. It's infinitely complicated. Some abnormalities characterize the genetic process. Some of these abnormalities involve whole chromosomes that do not separate properly during meiosis. When a gamete is formed, sometimes a sperm or ovum does not have its normal set of 23 chromosomes. For example, Down syndrome is a result of having an extra chromosome number 21, causing mild to severe intellectual abilities. In other situations, a person might have an extra X chromosome or an extra Y chromosome, or they might be missing a Y chromosome entirely. The treatments for these abnormalities do not necessarily erase the problem, but they may improve an individual's adaptive behavior and quality of life. Now, I don't expect you to memorize these abnormalities, but I do want you to understand that having too few or too many chromosomes can both result in abnormalities, some more severe than others. Abnormalities can be produced not only by an abnormal number of chromosomes, but also by harmful genes. More than 7,000 such genetic disorders have been identified, but most of them are rare. Here are some of the more common ones. For example, sickle cell anemia is a genetic disorder that impairs the body's red blood cells. Red blood cells carry oxygen to the body's other cells and are usually shaped like a disc. In sickle cell anemia, a recessive gene causes the red blood cell to become a hook-shaped sickle that cannot carry oxygen properly and dies quickly. As a result, the body's cells do not receive enough oxygen, causing anemia and often early death. If any gene-linked abnormalities run in your family, genetic counseling may be helpful. Genetic counselors provide their clients with information regarding the kinds of problem seen in this chart, the odds of encountering them, and also helpful strategies for offsetting some of their effects.